everyone's doing well.
I need to like set it on something.
All right. Is this
You'll need to
lighting
like
okay?
give like a water bottle.
I feel like
You'll
I
need
just
to like
made it
set it
very
on a water
sleepy.
bottle.
Maybe that's just me being sleepy. I don't know. Okay, it's good to see you all again. So I am here today to talk to you about neuropeptides, ACP, and other neurotransmitters. And so we have a couple of objectives, as we always do. We want to talk about neuropeptide action and how it's different from classical neurotransmitters, especially activity-dependent neuropeptide release. We want to talk about the mechanisms of purinergic signaling and the connection between adenosine and ATP. We want to explain the fundamentals of retrograde transmission, talk about volume transmission versus wiring transmission, and we're going to try to apply all this information to understanding experiments and proposed hypothesis and use this in the research lab. Now, just to give you a background on what this lecture is, okay, so you talked about GABA and glutamate and glycine and dopamine and all these other neurotransmitter systems. This is all the ones that you haven't heard about yet, okay? All squeezed into one lecture, all right? So what I've basically done here is I've kind of picked and chosen things that I think are gonna impact you in your research labs moving forward, okay? So this is really designed to be a lecture to inform you about what these different systems do, how it's possible to impact your interpretation of things, and to hopefully support you designing and doing things well and right the first time, rather than bumping into some problems that I've seen other people bump into, okay? So it's kind of, I don't want to say it's like a grab bag of stuff, but it kind of is, all right? So let's just get started. But just pay attention to the lecture objectives when you study, okay? So pay attention to the lecture objectives when you study, because there's a lot of stuff here. All right, so the first type of neurotransmitter or neuromodulator we want to talk about is the neuropeptides. Now, the difference between a neurotransmitter and a neuromodulator has really blurred over the last little bit. If you get an old neuroscience textbook, it will be very definitive about which is a neurotransmitter and which is a neuromodulator. But now that's kind of more relaxed, and it really depends on what field you're in and where your PI was educated and what they think what a neuropeptide or what a neurotransmitter or neuromodulator might be. And neuropeptides can sometimes be called neuromodulators, sometimes neurotransmitters. All right, so a neuropeptide is just a small protein, okay? It's just a small protein that's released by neurons and acts in a signaling way. And so as you learn from Dr. Lin, right, it's just a protein. It's got an N and a C terminus and peptide bonds, right there. So it's just a protein, okay? And the amino and the carboxy terminus are important as well. So neuropeptides are small proteins that serve neurotransmitter-like functions. They're really distinguished from other signaling peptides such as cytokines or growth factors, predominantly because of the receptors that they interact with and how they're released, although they can have overlapping functions. So just some examples of common neuropeptides that you may have heard of. There's the gut-brain peptides. This is cholecystokinin, galen, vasoactive intestinal peptide. The pituitary hormones can be neuropeptides, follicular stimulating hormone, pituitary adenylate cyclase activating peptide, oxytocin, the hypothalamic releasing factors, corticotrophin releasing factor, growth hormone releasing hormone, the opioid and related peptides, so the endogenous opioids are peptides, beta-endorphin, dynorphin, lumenkephalin, metenkephalin, endomorphins 1 and 2, and orphan and FQ. And then there's just other peptides, angiotensin, bradykinin, calcitonin, orexins, neuropeptide FF. So there's hundreds and hundreds and hundreds and hundreds, probably thousands of peptides. There's actually new peptides that get described about every year. Someone will find a new one. So this is an area of active growth. This is not an exhaustive list either. I'm not going to sit there and list them. And as you can guess from their names, right, oftentimes the peptides are named for what the people first described them doing, but they're in the brain too, okay? So, and doing things. So neuropeptides play a modulatory role in just about every neurological system, just about every circuit. There are hundreds of neuropeptides with unique functions, and they can have both local effects, and some are considered hormones, which really means they're released and they travel long distance to affect, other parts of the body, things like that. So they can do both. Oh, and they also make you look younger. Yeah. So this is this neuropeptide cream that you can rub on your face so that someone like me can look more like someone like you. No, I don't know if it actually works, but while manipulating the neuropeptides, neurotransmitters, and hormones that are in the brain to help create younger, more beautiful skin and slow the aging process, might seem more at home in a Michael Crichton or Robin Cook novel. Dr. Perricone says, believe me when I tell you it's not only possible, but it is in fact a reality. So I've actually had this slide in here for probably about 8 years, and I just now recently have realized this cream is like taking off in the beauty world. You can take brain peptides and rub them on your face. All right, so when we think about neuropeptides in the nervous system, the reason why we're talking about them in a different way is because they are synthesized somewhat differently than a lot of the neurotransmitters we talked about. So because they're peptides, they are synthesized, of course, from transcription, all right? So you get your pre-pro peptide hnRNA, which gets processed to our poly-A RNA in the nucleus, which gets transported out of the nucleus into the cytoplasm. You've already learned about this. So you get your pre-pro peptide mRNA that goes to the ER, the rough ER, where it hooks up with ribosomes, right? And here's the ER membrane, here's the ribosomes, here are the ER lumen, and as the peptide is transcribed for these peptides, right, they have a signal sequence that's just gonna say go into the ER lumen, right? So they get transcribed into the ER lumen, the signal sequence is clipped off by the signal peptidases, right, to make the pre-peptide here that is in the lumen. These pre-peptides then also get processed in a variety of different ways. Now, in addition to the processing though, you can also have alternative transcripts for these peptide genes, okay? So this is an example of the pre-pro-tachykinin gene A, and within this gene, you can actually transcribe multiple different types of mRNA. So there's the alpha PPT mRNA, the alternate transcript, the beta PPT mRNA, and the gamma PPT mRNA. So you can have 3 different transcripts, all from the same gene, so you can have that differential transcription. And then these transcripts will encode different peptides, all right? So this is really common when you have those pre-peptides, they actually have many different peptides within them. So for example, the alpha PPT can produce substance P, the beta PPT can produce substance P, neurokinin A 3 through 10, and neuropeptide K. And then the gamma PPT can do substance P, neurokinin A 3 through 10, and neuropeptide gamma, all right? So you have this incredible complexity in terms of what peptides can be produced. Now, once you have that signal peptide, or that pre-pro peptide or the pre-peptide, it can actually be processed differentially as well. So here is a signal peptide right here for the pro-opiomelanocortin or POMC, right? So here's that signal peptide that says go into the lumen. Right here is the protein that actually can encode for multiple different types of peptides within it. The signal peptide gets cleaved off, and then you have that pre-peptide, and then that can be processed by a number of different processing enzymes. So you have PC1 here that can process this protein into 2 different molecules here that can get further processed by PC1 and PC2 to ultimately result in the active peptides at the end there, right? Gamma MSH, alpha MSH. But this processing can also be tissue specific, all right? So you can have in the anterior pituitary, for example, predominantly the pathway stops here at ACTH. And when this transcript is made, when this protein is made, the predominant protein that's created is ACTH, all right? In this case, in the intermediate lobe of the pituitary, okay, then there's further processing that takes place, and the predominant peptides that are produced are alpha, MSH, and beta. Endorphin, okay? So you can produce multiple different peptides with this. Okay, so what does this mean if you're doing, let's say, transcriptome analysis of a region? All right, so you're gonna analyze the transcriptome. Does that tell you exactly what peptide is being produced in those cells? No, right? It tells you whether that particular transcript you're looking for, that particular transcript that you saw is there, right? Now, it's really cool actually. The processing of these peptides can also be changed by pathologic factors. So if you have a stroke or ischemia or something like that and you look in the brain, you have neurons that before the stroke might be producing predominantly one peptide, after it they might be predominantly producing another. Okay, so you can get this really, really cool regulation in terms of what peptides are being produced by cells, what peptides are being processed by cells that's dependent upon activity, environment, and a whole host of other things. Now, granted, all those peptides, they have to be in that prepropeptide, right? Like they have to be there, but that processing can be differentially regulated. And this is not something that is highly studied, okay? So it's not a big area of research, which always surprises me. But. Okay, so so this is just telling you the different types of peptidases that can do these modifications. So the peptides themselves also can have specialized modifications. So as they're being processed, you have the endopeptidases, which cleave within the peptides, and the exopeptidases, which cleave at the end of peptides. Some peptides also have disulfide bonds within them. You'll see though, I think we're going to talk about a couple of examples. So they actually have to have those disulfide bonds, giving them that nice 3D structure and stability that they need. And then there's also a whole variety of modifications that the peptides can have. So you can have an acetylated N-terminus. So this is a modification that occurs after. So you can have a peptide without the modification or with the modification. You can have an amidated C-terminus, all right, as well. You can have some other kinds of modifications where things are popped in there, some cofactors and things like that. So they can be modified post synthesis in some way. So the vast majority of neuropeptides as well, sorry, is this glitching too? No, it's okay, all right. So the vast majority of peptides, they're really produced in the nucleus, okay? The rough ER, and then they get transported down the axon to their site of release, okay? So think about that, right? Think about the neurons, right? It's being produced in the nucleus and transported down the axon, right? So when you have something like this, what we were looking at in this diagram here is it's showing kind of the difference between neuropeptide processing, where the vast majority of it is coming from the nucleus, down the axon, and the peptide is being processed all along the way as it's being transported down here to then be released at the axon terminal. And this is very different than those classic small molecule transmitters that we talked about, right? Those can actually be synthesized within this presynaptic terminal. The enzymes that do that synthesis might need to be created in the nucleus and processed down the axon. But then that one enzyme can produce thousands and thousands and thousands of molecules of neurotransmitter. When this process is used, right, you're having one molecule that's getting produced up here and has to transport all the way down there, and then when that gets released, you have to wait for more transport to come here if it's like that, okay? Now, you can get local translation in the terminal. I'm not going to talk about that currently, but that is something that can happen with a variety of transcripts. Have you talked about that already, that local translation in the terminal? So you can have some of that occurring, okay? But in that case, the RNA has to make it there. So, all right. So as this comes down, as this processing comes down, this is just another illustration of that, where you see, here's these peptides coming down the axon. They are distinct. Oftentimes they are packaged in vesicles that are distinct from your small, traditional, uniform synaptic vesicles, okay? So these are your dense core vesicles. So named because when they were doing those EM micrographs, they look dense. They look dense because they're filled with protein, because they're filled with neuropeptides, okay? And that is different from those small molecule containing vesicles that often are, relatively clear on those EM micrographs. And within one nerve terminal, right, one nerve ending, you can have both, right? You can have both those small molecule containing neurotransmitters and then the dense core vesicles. Okay, and then there's that illustration, right, of your dense core vesicles. Here's your EM micrograph, here's your presynaptic terminal here and here. Here's a terminal that has dense core vesicles in it, as well as the typical synaptic vesicles. Here's a terminal that does not have that, right, so you don't see any there. Okay, so now what this does is makes it really important when we think about neurotransmitter activity and neurotransmitter release. And I know when I talked to you about neurotransmitters briefly, I do think when I first started teaching this many years ago, actually I was teaching to med students, I inherited, did I tell you this story? I like to tell stories, I told you this story. I inherited a whole bunch of lectures about neurotransmission, right? And one of the things that one of them said in big bold letters is one neuron, one neurotransmitter. One neuron, one neurotransmitter, okay? That is not true. I mean, when I got these lectures, I was like, like, I don't know neuroscience at all. Like, what's going on? What's happening here, right? But it was dogma. So it used to be dogma that one neuron, if it's a glutamatergic neuron, it's going to release glutamate, right? GABAergic neuron is going to release GABA. It's not going to release something else. One neuron, one neurotransmitter. That is not true. Okay, so there's many cases where we have neurotransmitters and neuromodulators lining up together where neurons can release both, and that's what we're kind of talking about a lot today. Now, I don't know any cases of a neuron that's releasing both glutamate and GABA, okay? Like, that's not happening, so there's not much overlap there, but something that might be releasing dopamine and neuropeptides and maybe ATP as well and some other things, that does happen, okay? So, and with peptides, it's really important because the dense core vesicles release or fuse and release their content in a different way than the small molecule containing normal synaptic vesicles, okay? So this is an illustration of that. So if this is a presynaptic terminal, not a cell, it's a presynaptic terminal that's got both vesicles that are containing those classic neurotransmitters lined up on the active zone here, right next to your voltage-gated calcium channels. And then here are your dense core vesicles, and they're often kind of just a little bit away, okay? So they're not necessarily lined up right on the active zone, they're kind of in a distinct area, okay? And you can actually get frequency-dependent release of these two different types of vesicles, such that if you have low frequency action potentials, okay, nice low frequency, you're gonna get these local rises in calcium that are occurring around the area of those small synaptic vesicles, right? And that's gonna cause them to fuse and release their contents, right? Low frequency stimulation, we're getting the release of that one neurotransmitter. But then if you have high frequency stimulation, all right, lots of action potential, big train of them coming down, well, now we can have that same presynaptic terminal release both the small synaptic vesicle content as well as the dense core vesicle content. And this is due to a more widespread elevated calcium concentration that's occurring because of those repeated stimulations. They're happening so fast that we're not getting buffering, that calcium rise is being transmitted to a little bit farther away place, okay? And now we're seeing those vesicles fuse. Okay, yeah, and this release of these neuropeptides is a little bit farther away from the active zone, right? So this can be kind of non-synaptic sites that we're seeing. It's still near the synapse, but we're getting this sort of release there. Okay. Any questions so far? You are remarkably quiet today. Yes?
Sorry, the dense core vesicles, do they require the same like snare complexes?
It's different.
It's different.
Yeah, it's different. The more that they're studying it, the more differences they're finding. There are proteins in common, but then there's proteins that are different as well, and it really depends on the cell. So it's still kind of an active area of research as to why. You can imagine when Dogma said that you know, if you're a peptide secreting neuron, you're just a peptide secreting neuron, right? Like, it took a while for people to start studying these phenomena. So, all right. So with all neurotransmitters and neuromodulators, oh, go ahead.
I just wanted to ask, can the dense vesicles ever be spontaneously released?
You said, can they ever? So my inclination is to say yes, but I don't know if people like looking at that. It'd be really hard to tell, right? Because as you're gonna learn, a lot of all the receptors for the neuropeptides are metabotropic receptors. So these are a little bit slower acting anyway, so it's hard to look at those mini events. You know, I talked a lot about mini events with you. Those are usually used when you're looking at GABA and glutamate, not so much for other things, just because the responses are so slow. So I imagine they can. There's probably some studies out there that they do, but I don't. Yeah. All right, so what happens after the neuropeptide is released? So neuropeptides as a whole are not rapidly cleared from the synapse, they're long lasting. And so oftentimes they can kind of just diffuse away and do what's called volume transmission, which we'll talk about in a bit. So they just sort of leave the area, right? They can be degraded by extracellular endopeptidases and exopeptidases that will cleave those peptide bonds and create an inactive peptide. There's no evidence yet for peptides being taken back up and used again. So it's kind of like one shot and done. They release, it's done, it's out there, it's having its effect, but there's no evidence of recycling that's happening there. Oh, here's the volume transmission and what it is. Okay, so volume versus wiring transmission, and this is one thing you see in a lot of systems. So what you're looking at here, these are two different neurons. There's one neuron here and one neuron there. And and this one's got an axon coming down here, right? That's forming a presynaptic contact with this neuron that's got an axon coming down here, forming contacts with that axon, okay? And so if we look at this axon right here and we think about these presynaptic boutons releasing their neurotransmitter right along here where you've got a defined postsynaptic area, right? That's wiring transmission. So we've got a defined presynapse, a defined postsynapse. They're sort of wired together, okay? We can also have volume transmission in that sometimes we will have a presynaptic bouton that does not have a postsynaptic response or doesn't have a postsynaptic element next to it, right? And so you can get release of neurotransmitter that just kind of floats away, or it's being released from a presynaptic terminal that does have a postsynapse and is diffusing away and out, okay? That's called volume transmission, right? These terms kind of fell out of favor for a while, but I'm actually starting to see them pop up in the literature again, this volume transmission and understanding that. So, and you can have the same axon do kind of the same kind of thing where you can have wiring transmission and volume transmission from the same axon. This is particularly true for peptides, okay? We see a lot of that with peptides. All right. Okay, so as I alluded, all neuropeptide receptors are metabotropic in vertebrates and mammals, all of them. So there's no ion channel that the peptide binds and causes it to directly open. All right, peptides can modulate the activity of some channels, but there's no like peptide gated channel. So all of them are metabotropic, G protein coupled receptors or other types of signaling receptors, okay, cell signaling receptors. So that really means that what's happening is the peptide is binding to that G protein coupled receptor, which is a 7 transmembrane domain. When you look at the topology, it kind of looks like an ion channel, but it isn't, doesn't have a core, right? So the agonist binds to the G protein coupled receptor and causes a conformational change in the cytosolic region of these receptors that cause activation of signaling cascades, right? So this is an example of how these G protein coupled receptors work. So here's the receptor itself, here's the agonist, and generally the GPCR is hooked up to a G alpha protein, a G beta protein, and G gamma protein, which is bound to GDP. This is sort of in its receptive form. When the agonist comes in and binds, there's a conformational change which activates these G proteins and releases them from the receptor. So they go off now, and this activated alpha protein can go hit target proteins and initiate a whole variety of different signaling cascades, and then the beta and gamma subunits as well can shoot off and go interact directly with proteins to modulate their function. All right, so have you had the lecture on G proteins? I'm seeing some nods and yes, okay, so we have had some talk about this.
I think Overtime talked about it a little bit.
That's what I thought.
Not
They
really
got mentioned, but
formally.
we've never had like a specific like lecture. on it.
Okay, so it got mentioned.
That's right.
All right, okay, good. I thought it was getting mentioned, but I wasn't sure. So this is sort of the pathway for how this works, right? Let's see, do I have more? Yeah, okay. All right, so this is the pathway for how this works. There's different canonical pathways as well with this. But the really important things to know, right? This is just another illustration of that G protein coupled receptors and how they work. Important things to know is the GPCR itself is not an ion channel, okay, right? But these activations of these G proteins can come off and affect second messenger cascades that can adjust and alter the activity of ion channels. So it can cause ion channels to open, it can cause ion channels to close, right? And it can modulate their activity, generally through phosphorylation or association of those beta and gamma subunits. All right. The ion channel is physically separated from the receptor, although they may be in close proximity. Let's see. Oh yeah, and when we're talking about these mechanisms of action, again, they generally have a slower onset and a slower offset, okay? It's built for duration and complexity, not for speed. And G protein coupled receptors really are the largest protein family in the human genome with over 900 different members. For those of you who are thinking you might want to go into industry and start to do like therapeutic drug design or talk about drugs, these are a huge target. Okay, so understanding how these work, this is a huge deal when you start to get into that pharmaceutical area. So here's just an example of how these signaling cascades from the G protein coupled receptors work. All right, so you'll have that external signal in the outside environment that's going to interact with the G protein coupled receptor. And then the G protein coupled receptor is going to have a primary transducer that's going to interact with a primary effector that then goes off usually and affects the second messenger system and causes a secondary effect. All right, so an example of this, here's the cyclic AMP dependent system. So norepinephrine can interact with the beta adrenergic receptor, which can act through G alpha S for stimulating, which can stimulate adenylate cyclase activity, increasing cyclic AMP in a local area of the cell, activating cyclic AMP dependent protein kinase, which can then go off and phosphorylate things and change their activity, all right? Here's one that depends on the phosphoinositol system. Acetylcholine can interact with muscarinic acetylcholine receptors that act through GQ to stimulate phospholipase C to produce IP3 and diacylglycerol. IP3 can go off and do some calcium release from ER stores, and diacylglycerol might activate protein kinase C that will go off and phosphorylate proteins, all right? So whenever you have activation of G protein coupled receptors, you can have sort of those effects that you might see if you're doing neurophysiological analysis, right? But there can be other effects inside the cell as well, as you can imagine. So it can get very complicated. Now, when you think about neuropeptide systems too, some neuropeptides, such as thyrotropin releasing hormone or whatever, bind to only one known receptor. So there's a peptide and a receptor, and they both have the same name, like this is named peptide and this is named receptor, and you know the two go together. But some neuropeptides can interact with as many as 5 different neuropeptide receptors. Some of them do not have the name of the peptide in their exact name, okay? So whenever you have peptides produced, you gotta kind of think a little bit outside the box and look at the literature a little bit to see if there are other target receptors beyond the one you're thinking about, all right? Now, neuropeptide receptors themselves can bind more than one neuropeptide as well, and some of them don't necessarily have all the same name, right? So you can see that. Neuropeptide receptors themselves will show synaptic and extrasynaptic locations. So sometimes they're just peppered across the cell body. Sometimes they're in a primary cilium and the synapse. So you can see these receptors in different locations, which makes sense when we're thinking about like volume transmission, right? So if the peptide's kind of coming out, you might have receptors in many different areas to detect whether it's there or not. There can be some difficulty in studying some of these systems. There is anything that was described probably within the last 10 years may or may not have defined forms like drugs or compounds that can inhibit or activate it beyond its endogenous peptide. So there can be a lack of available agonists or antagonists for some of these things that really slow things down, okay? Same thing with antibodies. Finding validated antibodies for working with them can be a bit of a challenge. So most peptides got their name because of either how they were first described, if they were described in a certain system to do a certain thing, or if they were a neuropeptide. Oftentimes what they did is they did an intracerebroventricular injection of the peptide into an animal, so into the ventricles, right? At a high concentration, and then watched to see what the animal did, okay? So the first order of understanding of a lot of these peptides might have been somewhat off, right? Because if you inject a high concentration, you might see an activity or a behavior that maybe that peptide doesn't normally do. Given that now you all know all these different things, right? So injecting a high amount of a peptide in there, who knows? So oftentimes you need knockouts or disruption of individual receptors to really know what the peptides are doing. Then usually the agonists and antagonists follow afterwards, but there's a delay there. Okay. All right, does anyone know why there's a delay these days? So where do most of our agonists and antagonists come from? The good ones, at least. Do I, as the PI, create those? Really not, right? It's medicinal chemists, pharmaceutical chemists, and companies, right? So companies might identify something. So if a company identifies a new agonist, do they immediately release it to be sold to scientists? No, right? They hold on to it because they're patent rights, right? So they start releasing it, the clock starts ticking. So they'll often hold, okay? So that kind of slows things down, and we see that. And that's okay, that's just the way the system works. But sometimes you can actually find agonists or antagonists to use in your studies by getting your PI to talk to a company to try to see if you can get access to things that you can't get from a catalog. Okay, all right, you're all still with me, this is good. Okay, so one of the examples that I just want to talk about really quick, because we want to talk about one thing that's really important, is the endogenous opioid system. So opioids, right, the term opioid refers to compounds found in opium. Okay, we know of these for their pain reducing effects as well as their propensity to cause addiction, right? So we need it, it's useful, but yet it can cause these addictions. They have several adverse effects. So there's been a lot of study of the endogenous opioid system, right? We want to know what's going on because we want to use the system to prevent pain, but also not have addiction. So this is just an example of the different opioid peptides and the different opioid things. We have the mu opioid receptor, that's the analgesic, the euphoric, the addictive one, that's the one that a lot of people target. We have the kappa opioid receptor, which has the dynorphin peptides, have the highest affinity for that. And we have the delta opioid receptor, and then we have the orphanins as well. Now, even though the mu, let's say, beta endorphin interacts with the mu receptor and endomorphin interacts with the mu receptor, that does not mean that dynorphins at high concentrations can't stimulate the mu, right? So if you're doing a study or something like that and you decide what concentration of peptide I'm going to use, you go too high, you are going to hit them all, guaranteed, okay? So finding that right concentration is really, really important. Now there's another thing as well that happens that originally really became very well understood with the opioids, and now we're starting to see it applied to a lot of the other G protein coupled receptor pathway. And this is this concept of biased agonism, all right? So when I talked about G protein coupled signaling, and when Dr. Oberton likely talked about it, we talked about this canonical pathway, this G protein mediated signaling pathway. Now, here's an example. Here's a G protein coupled receptor right up here. Here's its agonist. It's normally coupled to an alpha, beta, gamma. Okay, when the agonist binds, these go off and have actions. Now, there is a canonical pathway for most GPCRs that say, oh, it's going to work for G alpha S or G alpha Q or whatever. But most of the G protein coupled receptors, particularly when we start to study them in neurons, we find that in different cell types, they can couple to different G alpha proteins. So for example, this one can go G alpha S, G alpha 12, 13, G alpha Q, right? So they can couple to different ones depending upon the cell type, and depending upon the alpha protein that's coupled to, you can get different actions such as raising cyclic AMP, lowering cyclic AMP, activating the Rho-Jeff situation, activating the phospholipase B or C, okay? So you can get different effects with that depending upon what, sorry, my screen just went back, depend what it's coupled to. Now, the other thing we now know that happens, so G protein coupled receptors, after they're activated, become desensitized, okay, by these GERK proteins that come in and phosphorylate the GPCR. This then gets bound by proteins called arrestins, which then cause the internalization of the G protein coupled receptor into these clathrin-coated pits, which then moves the G protein coupled receptor to the endosome, okay? And this really used to be thought to be, then it goes to the lysosome where the G protein coupled receptor is degraded, or the endosome will recycle back up to the plasma membrane. So this was thought to be a desensitization cascade for a long time. G protein coupled receptors desensitized by being internalized. However, we now know that in the endosome here, when arrestin is bound, we can get arrestin-dependent signaling. And so this is another signaling system that gets activated where it can activate ERK and other pathways within the endosome and then affect cellular processes in this way. So with G protein coupled receptors, you can have both that G protein dependent signaling as well as that arrestin dependent signaling. Okay, so you can get both of these pathways that get activated. All right, now biased agonism, and what is it? Both of these pathways are not activated equally by every ligand. Okay, so this is illustrated in this. So in here, we're going to look at what a biased, let's look at what a biased receptor is first. That's easier to understand. So in a biased receptor, here's a G protein coupled receptor here, and an agonist binds to that G protein coupled receptor, and maybe this receptor predominantly acts through that arrestin pathway. Doesn't mean it doesn't do G protein coupled signaling, but it does a lot of arrestin dependent signaling, okay? The same agonist could interact with another receptor, okay? And maybe now you get a lot of G protein coupled receptor signaling and not a lot of arrestin dependent signaling, okay? So that's receptor dependent. Some receptors are going to be more aligned to go one direction versus another direction. Biased agonists, however, will interact with, one agonist will interact with this receptor here and trigger more beta arrestin signaling, whereas a second agonist can interact with the same receptor as up here and trigger more G protein coupled receptor signaling, okay? And so this is widely occurring. It had a little bit of difficulty getting through to the mainstream because it was thought that G protein coupled receptors for a long time acted, and other receptors, it was like a lock and a key, right? Like every receptor had one lock, you just turned the key, the door opened, and that was it, the door just opened, right? But that's not true. It's not a door, right? Like it's a protein. And so you can get different types of conformational changes depending upon how different ligands will bind, all right? And that can trigger whether you're going to be predominantly G protein based or beta arrestin based, okay? Now the trick of this is that different ligands do different things, okay? So you can so easily, let's say you decided you wanted to look at what would happen if you activated a particular G protein coupled receptor in your model, right? or an antagonist or something like that, and you go to the catalog and you grab something, you might grab a biased agonist by mistake, right? So a lot of the biased agonism and the results from that's been pretty recently described. Some of it is the catalog might say this is an agonist for this, but you go and look in the literature and like, oh, that only activates the G protein signaling and not the beta arrestin signaling, right? So it's important to kind of know that that exists. This has been really important for drug design in particular because with the opioids, they've been trying to target certain systems hoping that they can get rid of certain bad effects and keep certain good effects by what targeting system that they're, by what the agonist is doing. And so if we had a non-biased ligand, it would activate both equally. If it's G protein biased, it's gonna be G protein biased. If it's arrestin biased, it's gonna be arrestin biased, right? And so we'll see a lot of that with the opioids where they really, really want to, let's say for example, we're targeting the delta opioid receptor here, and we know that if we act through these G-alpha I pathways, we can get analgesia, anti-Parkinson's effects, and antidepressant effects. But if we act through the beta arrestin pathway, we can get seizures, right? So we really want an agonist that's gonna particularly affect this and not that. And so we see a lot of that kind of thing going on with drug development, drug companies, and things like that. All right, yeah, and it's not just for opioid receptors, okay? So think about this if you're starting to target a new system that your lab hasn't worked in or things like that. It's important to do a comprehensive analysis so you don't step in it and then go and publish your paper and you send it in and the reviewers say, yeah, but you used that. You gotta use this. And then you gotta repeat all your studies all over again to publish it or completely change the focus of what you thought your project was about, right? So, okay, all right. Okay, we good. Any questions about neuropeptides and how cool they are? They're very cool. I like neuropeptides. All right, okay. So let's talk about the purinergic system and how it's tricky. So purines and purinergic signaling occurs throughout the body, right? Or it's important throughout the body. This is just what a purine is. Here's a purine, here's the adenine, here's the guanine. Yes, you're thinking about things right. These are the building blocks of DNA and RNA, right? They're also metabolic cofactors that you learned about. They cause second messenger systems, cyclic AMP, cyclic GMP that we just kind of talked about. The nucleosides and nucleotide derivatives of the purine bases are also neurotransmitters, and they play a very important role in signaling between neurons, okay? So they do all of these things, and they're neurotransmitters. Yep?
Do they do that in these exact forms, or are they modified a bit?
ATP is a neurotransmitter.
Is that it?
Yep, and so is adenosine. Yep, ATP is a neurotransmitter. It's a very powerful one, yeah. So here's the purinergic signaling. So here's our ATP molecule right here. So here's the adenosine right there. Here's the adenine, right? And here's our ATP with the 3 phosphates, 1, 2, 3. If you only have one, you're AMP, 2, ADP, 3, you're ATP, okay? So when we're thinking about the principal neurotransmitters here, they're usually ATP and adenosine, but there's also another molecule that it's very rarely studied. When it pops up, it's usually some big paper and then it sinks back down, disappears from people's consciousness. But the adenine dinucleotides, they're generally called APNA, with N being the number of phosphates, and they're basically 2 of these hooked together with phosphates in between, and they can have like 11 or 12 phosphates between them, okay? So those can also be found in synaptic vesicles, all right? This is AP4 with 4 phosphates there. Okay, so ATP and the APNA are actually stored in small synaptic vesicles right alongside glutamate and released in response to action potentials in that calcium-dependent process. So again, we're eroding that concept of one neuron, one neurotransmitter. There's ATP there, okay? So ATP is a classic neurotransmitter and is often co-localized in the same vesicles. Adenosine is a little different. So adenosine can be released from non-vesicular cytoplasmic stores and reaches the extracellular space through transporters or ATP metabolism. We're gonna talk about that in just a minute. And that is when ATP is released into the synaptic cleft, there are things there that take the ATP, degrade it, and convert it to adenosine very fast. So whenever you have ATP, you have adenosine. They go side by side, okay? So that is just really illustrated here, where we have an ectodiphosphohydrolase on the presynaptic terminal of most presynapses. When that ATP is released, it gets converted to ADP, AMP. There's also an ecto 5' nucleotidase that converts that AMP to adenosine, and away we go. So whenever ATP goes out, adenosine's there too. And ATP goes out a lot. Okay, so just like any classic neurotransmitter containing system, we have receptors for ATP. We have the P2X receptors. These are ATP-gated ion channels. They're permeable to both sodium and potassium. So when we open a sodium-potassium channel at rest, what kind of response do we get? Oh no, you make me, come on. If I'm gonna open a ion channel permeable to sodium and potassium equally at rest, at the resting membrane potential, what is going to happen to my membrane potential? It's gonna depolarize, right? So these are depolarizing. The P2X, yes, it's classic depolarizing. Sodium comes in, potassium goes out, right? But sodium comes in, it looks just like a glutamate receptor actually, except it's gated by ATP, right? So that's a classic receptor. We also have P2Y receptors. These are G protein coupled receptors activated by ATP, right? So they work through the second messenger systems in that pathway that we just spent a lot of time talking about. So those are the P2Ys. P2Xs are the ion channels. The P2X, this is just an aside because I think it's cute. I like ion channel structure, activity relationship type things, and when the crystal structure of all these ion channels came out, I was really very excited about it. And this one happens to be my favorite, even though I don't work on it. Because when they solved the crystal structure, so most of these multiple subunits come together to form the functional channel. This is no different. The channel has a pore down the center, no different. But when you look at the individual subunit, here's the transmembrane domains. It's got this big extracellular region with a lot of disulfide bonds in it. But when they solved the crystal structure, they likened it to a dolphin. So that is down there, this little dolphin. So this is the fluke region, and it will be forever called the fluke region. This is the dorsal fin, the right flipper, left flipper, the flipper, body, and the head. And there's a little picture of why they think it's dolphins. So the full channel is like 3 dolphins, like together. All right. Okay. So there's that. The adenosine also has its own receptors. These are G protein coupled receptors. There's adenosine 1, 3, 2B, and 2A. 2B and 3 are not highly expressed in the brain, but the 1A and 2A are. When adenosine can interact with these, they can affect G protein coupled regulation, and oftentimes the 2 different adenosine receptors sort of antagonize themselves a little bit. So usually there's one more in the cell than another, okay? So this is just kind of showing their canonical pathways. One set might inhibit adenylate cyclase activity, one set might excite it, okay? And then they'll cause changes in ion channel activity. So how might this knowledge apply to you? So why do you care about the purinergic signaling cascades compared to others, okay? So one of the things is it's tricky, okay? So it's a little tricky because whenever you have things being released, you're looking at release, you think you're looking at release, you've got ATP going there, but you've also got adenosine. So the number of papers that I've seen that have had to been corrected because someone thought they had an ATP dependent effect when it was an adenosine effect are very, very high. In fact, there are a whole number of things that we need to do in the electrophysiological core when we start to slice slices, right? We get some neurotransmitter release. We do all sorts of things to prevent glutamate from doing bad things to the slice. But there are a whole host of experiments, we have to go in there and inhibit the adenosine receptor, right? Because that actually will get activated during the slicing event and cause all those signaling cascades to occur. And if we want to see a certain thing, we have to make sure that that's inhibited even when we slice, okay? So it's a kind of a sneaky system that you gotta know it's there. All right, questions about that? Okay. All right, retrograde transmitters. Okay, so far we have talked a lot about a presynapse releasing a neurotransmitter that diffuses away and affects a postsynaptic response, right? We talked about that. And generally for a long time, it was thought that that's the way synapses worked. So here's a presynapse right here, here's a postsynaptic membrane. We get a signal in that presynapse, right? It comes down and affects the postsynapse. For a long time, it wasn't thought that the postsynapse could then come back and affect the presynapse, right? Like everything was unidirectional. Everything was pre, post, and then that one went down and pre, post, right? Everything is going in one line. So what a retrograde messenger is, okay? A retrograde messenger is where we can have activation of the postsynaptic element, and then the postsynaptic element itself can respond and come back and affect the activity of the presynaptic element directly. So this is retrograde synaptic signaling, where we're going from post to pre, not pre to post, okay? Now there are a number of different molecules that do this. They're called retrograde transmitters. It's growing as to who can do this, but growth factors will do this. Adenosine, which we just talked about, will do this. The gases, nitric oxide and carbon monoxide. Yes, carbon monoxide is endogenous in your brain at really, really low levels, okay? Nitric oxide, carbon monoxide, and then also the lipid-like molecules, endocannabinoids, all right? So all of these are retrograde messengers. So we're gonna talk just briefly about the nitric oxide cycle, just briefly, I hope. Quick. So nitric oxide is best known as a potent vasodilator, right? There's multiple medicines out there that enhance nitric oxide dependent signaling, including Viagra and nitroglycerin, right? So this has been used forever. When you talk about nitric oxide, do not confuse it with nitrous oxide. Nitrous oxide is the stuff that you breathe at the dentist office that makes you happy, all right? It also is a drug of abuse, okay? That is not the same thing as nitric oxide. This is nitrous oxide, this is nitric oxide, okay? So nitric oxide has its own defined signaling cascades. These can be particularly important in neurons, so bear with me. This is a presynaptic element here, this is a postsynaptic element here, and then we have a glial cell that's around the synapse or near the synapse, and we actually have another neuron here as well, okay? That's just hanging out. And so the cycle as it goes, and this should remind you of something, okay? So the first step that happens to get nitric oxide retrograde signaling is that you have a rise of calcium in the postsynaptic cell, all right? So there's some sort of rise in calcium. This can be due to opening the NMDA receptor channels, right, that have calcium permeability. Or it could be from opening voltage-gated calcium channels that happen to be in the postsynaptic membrane, or it could be due to release from intracellular stores in response to activation of a G protein coupled receptor, right, that we just talked about. So you get this rise in postsynaptic calcium, right? What does this remind you of already? You've had a lot of lectures, tell me. Plasticity, okay, that's good. What else have we talked about?
Transmitter release?
Okay, transmitter releases on the presynapse, right? Have you talked about calcium-dependent signaling cascades yet? Right? So there's a whole host of calcium-dependent processes. Okay, so yeah, so this is just one of those. This is additional one of these. You haven't talked about this yet, right? Okay, all right. Okay, so calcium rises in the postsynaptic membrane, or the postsynaptic cytosol there. That's going to activate this enzyme, nitric oxide synthase. So nitric oxide synthase gets activated by that rise in calcium. In this case, it's called nNOS N-nitric oxide synthase. The N stands for neuronal, right? NNOS. That's gonna use arginine to produce NO, all right, and citrulline, but we don't care about that. So arginine produces NO, that's gonna be that nitric oxide, that gas. And then because it's a gas, it's gonna go through biological membranes. It cannot be contained, it's just gonna go, all right? So it's gonna go and it's gonna diffuse across membranes to then influence other neurons, other cells, and glia, okay? So once it's produced, the NO is produced here, it just leaves, or it stays, okay? But it's gonna activate soluble guanylyl kinase and cause an increase in cyclic GMP within the postsynaptic cell, or the glial cell, or the presynaptic terminal, or the other neuron that's nearby it, okay? Because it just diffuses out, okay? And that can cause a whole host of signaling cascades, particular activation of protein kinase G, which can go off and phosphorylate a whole number of different proteins, including ion channels, and affect ion channel activity, okay? All right, so it can do that. Now, NO can affect protein activity in a couple of different ways, ion channel activity in a couple of different ways. So it can do it through this PKG mechanism, but it can also cause nitrosylation. So nitrosylation is basically a direct covalent bond at free cysteine residues in the protein. So that's kind of shown here. Here's the NO, here's the free cysteine, and you can get that NO bonded right to the cysteine. And of course, if you have that covalent bond on that cysteine, that can cause a conformational change or affect how the protein undergoes conformational changes to affect its activity, okay? And there are a whole host of proteins in the nervous system that are known to be nitrosylated by NO, okay? This is showing the voltage-gated sodium channels, right? Responsible for the upswing of the action potential. The L-type calcium channel, these are those large, long-lasting ones, not necessarily the ones that are involved in the neurotransmitter release, although those can be affected as well. The NMDA receptor is sensitive to nitrosylation. It changes its gating. Okay, so there's a whole host of things that can be affected by the modification of these thiogroups on these proteins. Okay, all right, so that's the NO system. The NO system exists, it's there. There are very specific things that you can do to target it. You can inhibit the enzyme activity, okay? That NNOS, you can get things that inhibit it, you can get things that'll activate it. So there's a whole ways to look at it. The key part here is this enzyme, this NNOS enzyme that produces the NO, that is the rate limiting step. If that is not there, NO is not produced, okay? So oftentimes what you'll see is if you have, let's say, you're looking at, let's say, a slice of a brain or a big area, you might have a whole bunch of neurons that do not have NNOS, and then you've got one that does, right in the middle. Okay, and then that one, when it gets activated, produces NO that diffuses out and can affect all these other cells. Okay? All right. Okay. All right. Questions about that? Okay. All right, so the next one we want to talk about is the endocannabinoids. Okay, so the endocannabinoids are the endogenous cannabis cannabinoid system that we have. There are two predominantly endocannabinoids. We have anandamide, which was the first one described, and then 2-AG, which was the second one. And these, again, are the endogenous molecules that underlie the action of THC and cannabidiol. And these are considered retrograde transmitters. So there's also two known cannabinoid receptors, CB1 receptor and CB2 receptor. Both are G protein coupled receptors, okay? So they are G protein coupled receptors. CB1 receptor is the most abundant GPCR in the nervous system. I'm gonna say that again. The CB1 receptor is the most abundant G protein coupled receptor in the nervous system, okay? So there is a ton of this. The CB2 is present in immune cells in the immune system, but not necessarily in the neurons and glia of the brain. CB1 receptor is also often localized to the presynaptic nerve terminal and is a potent inhibitor of synaptic strength. Let's talk about that. So this is just a schematic of what you see. Here's the presynaptic terminal, here's the post here, okay? And just like we saw with NO, it's a slightly different mechanism, but we have this cannabinoid receptor that's here that can affect presynaptic activity, but it has to get produced to do that, right? And so here you're looking at neurotransmitter release when vesicles fuse. You have neurotransmitter that can go out and activate different neurotransmitter receptors. What signals all of this is a rise in intracellular calcium, yet again, okay? So a rise in intracellular calcium is going to cause the production of the endocannabinoids, which can then release, can be released. Okay, I'm sorry, I'm looking at this and I'm like, okay. I changed this to simplify the lecture for you this year, and so there's some like cues aren't present. Okay, so there's the rise in calcium. Now, one of the things that's important to consider here with endocannabinoids is that they are lipids. Okay, they're lipid-like, they're hydrophobic, and so they also cannot be contained. You can't put it in a vesicle and then have it be released because it can't be contained. Okay, can't be contained there. All right, so once it gets produced, it's released and it goes off and it does its thing. And generally what activation of the CB1 receptor will do in a presynaptic terminal is it has a variety of effects. And that is predominantly that it will, working through G proteins, it will inhibit voltage-gated calcium channels in the presynaptic terminal. What's that gonna do to my neurotransmitter release?
Decrease.
Decrease it, right? It will also open a potassium channel. Okay, and this is not the delayed rectifier potassium channel, this is a potassium channel. So what's that gonna do to the presynaptic terminal?
Hyperpolarize it.
Hmm?
You said potassium.
Yep.
Hyperpolarize it.
It's gonna hyperpolarize it, right? And so what's that gonna do to neurotransmitter release?
Decrease it.
Decrease it, right? So the presynaptic membrane is gonna become much more hyperpolarized. You're gonna have some inhibition of your voltage-gated calcium channels. Together, these are really gonna dampen down that response, okay? And so with that, when we're talking about the endocannabinoid system, we can see that we get production of the endocannabinoids in that postsynaptic area, right? That's what we're seeing here. We have a rise in calcium that can activate phospholipase C, that can produce diacylglycerol that works through diacylglycerol ligase to produce 2-AG. And then the 2-AG can diffuse out, okay, interact with the CB1 receptor and inhibit presynaptic transmission of that same synapse, okay? In some cases, you have a system where all you need to do is depolarize that postsynaptic cell and the endocannabinoids are produced. And they go out and they inhibit that presynaptic activity right there, okay? And that inhibition can last for minutes, right? It's a G protein coupled receptor. We can also see situations, there's multiple circuits in the brain too, where we have a presynaptic activity, right? We might have activation in this synapse of the NMDA receptor or activation of the metabotropic glutamate receptor that can stimulate production of 2-AG, which then diffuses out. And this synapse in itself might not have a CB1 receptor. So if it doesn't have the receptor, then it's not going to be affected by the endocannabinoid. But a synapse near it might. So what this is showing right here is a CB1 receptor on a presynaptic GABAergic terminal. So this is a different cell, but we're getting release of this 2-AG that interacts with the CB1 receptor and can inhibit activity of this presynapse right there as well. And so there's many examples of that in addition, okay? In the brain in particular, 2-AG is really the most abundant. The anandamide was described first, but most publications that you look at, they have to prove whether whatever effect they're seeing is anandamide or 2-AG. It's usually 2-AG, okay, that's doing this, okay? And these are really, really very, very potent inhibitors. And I mean you know, taking something from, it's 100% down to 20% very, very quickly, okay? And then leaving it at 20% activity for a long time. Okay, all right. So really what we've talked about is how neurotransmission can be a little different, a little strange, right? We've talked about intrasynaptic vesicle release and spillover of the neuropeptides that can diffuse away. Okay, we talked about how peptides can be released at non-synaptic sites, okay, kind of diffuse away. We talked about non-junctional varicosities, where we have a presynaptic bouton and we don't have a postsynaptic response, that can diffuse away. Okay, we haven't talked about the carriers, but we've also talked about the gases and how once those are produced, nitric oxide's produced, it can diffuse away. And then also the endocannabinoids, once produced, they can diffuse away. Okay? So do we have any questions? I ended early somehow. Very quiet today. All right, so we know how to apply all of this? I think the thing is just be able to know that this stuff exists, being able to know that it's out there, and be able to know that you as a scientist can investigate this, right? And sometimes it's important as a student to think outside the box a little bit, right? And make sure that what you're doing and what you're targeting and what you're seeing is in fact what you think. I'm just gonna tell a story since this is not in here. I think I was, anyway. So there was an effect in the cerebellum that for years was ascribed to metabotropic glutamate receptors, okay? This was kind of a big deal. Metabotropic glutamate receptors do this thing, they do this thing, they do this thing. And then someone put in an inhibitor of CD1. And the metabotropic glutamate receptor initiated the production of the endocannabinoids. And then the endocannabinoids were the actually business end that was doing all of the effects. And they actually had the metabotropic glutamate receptor in the wrong location. It was on the post-synapse, they said it was on the pre. Everyone was ignoring the evidence that it wasn't actually on the pre-synapse because they could see these effects. And yeah, so that was like a big turnover when that one happened. Okay? All right, thank you for your attention.
Thank you.
Oh, and I have this, the SEI review. If you can, please do so. I'll make sure, I think, yes, this one's in there. Please do so. I am an associate professor, not a full professor. So what you say does matter in terms of whether I get promoted or not. So if you can, please do it. We have a really big problem with this class, even though there's 25 of you. Often so few do it that we can't actually use the SEIs, which please do it, okay? Because if it's below a certain level, we're not allowed to put it into our dossier. So, okay, thank you.